This is your host, Alice Formiga from eOrganic at extension.org. All eOrganic webinars, as well as many other resources on organic seed production, are archived on our website and on the eOrganic YouTube channel. This presentation will last about 45 minutes, and then we'll have 30 minutes for questions. We'll be reading as many questions out loud as we can after the presentation is over. Before we begin, I'd like to introduce our speakers. All of today's speakers are members of a national NIFA OREI-funded research project called the Tomato Organic Management and Improvement Project. First, we'll be hearing from Lori McKenzie, who is a Research and Education Associate at the Organic Seed Alliance. And then we'll hear from Julie Dawson, who's an Assistant Professor in the Department of Horticulture at the University of Wisconsin-Madison. And after that, we'll hear from Dan Eagle, who is a Vegetable Pathology Extension Specialist at Purdue University. So thank you, everyone, for joining us today. And I'm going to hand over the presentation to our first speaker, Lori McKenzie.
Okay, great. Thank you, Alice. Good morning, everyone. All right, it's working. I'm going to start out with just a basic overview of what we're going to talk about today. I'm going to start with some terminology, most of which I'm sure you will already be familiar with, but I just want to make sure that we are all on the same page and understand definitions in the same way for our discussion today. And then I will be talking about some genetics and how the genetics play out in a self-pollinating crop, which will help you understand some things as you go through your seed saving and seed production adventures. And then Julie's going to talk about choosing varieties and managing field plants, as well as genetic maintenance and harvesting of the seed. And then I will come back and talk about some seed processing. We'll be talking about fermenting tomato seed. And then Dan will wrap up with some disease prevention discussion. So the first terminology, we're going to start very basic, open pollinated versus hybrid. You will see both open pollinated and hybrid often referred to as F1 varieties in tomatoes. Technically, when you're speaking biologically, tomatoes are not actually open pollinated because they are self-pollinated. So they're not openly pollinating with other plants, they're just pollinating themselves. So leading straight into that, self-pollinated being single plants that pollinate themselves versus our cross-pollinated crops, which openly share pollen among multiple plants in the population. And these two flower shots are to show you that not all tomato plants are always self-pollinated. And one of the visual structural cues that you can use to determine if you've got some more likely to outcross or cross-pollinated tomatoes, this flower you see here on the left, this is the stigma. Inside, there's actually a fused anther cone that surrounds the stigma, and as the stigma grows and the flower matures, that stigma pushes through the anther cone, getting this very, very sticky top of the stigmatic surface, pushes through that pollen cone, so you get ensuring self-pollination. Over here on the right, you've got a tomato flower, and you may even find flowers like this on the same plant. It's not necessarily that all the flowers on a plant will look like one or the other of these, but you can see this is a very different structure in that the style and the stigma are now pushed outside of that anther cone. So now this stigmatic surface is available for pollen from other plants. So something to keep an eye on as you're going through and looking at your plants and thinking about your seed production. One more level into the terminology is phenotype and genotype. So the phenotype is what you see, and the genotype is what you don't necessarily see, but it's the genetic makeup of the plant. And F1 is probably the most familiar to you and likely what you see mostly out in the world, but F2 and F3 and F4, that's a denotion for a generation. So the F stands for filial, and it indicates the generation after a cross or after a hybridization. So the F1 is the first generation after a cross. And what you see, the phenotype, so when you're going through your plants and looking at through all your fruits in the field, sometimes you're going to see something that might look spectacularly different, and that is sometimes genetic and sometimes not, or it's possibly a combination of the environmental influence as well as the genetic makeup of the plant. So you want to keep this in mind because the environment and the production system will have significant impacts on the phenotype. And this is why I'm going to talk to you a little bit about genetics so that you can maybe piece some of this out and hopefully get a deeper understanding of how this happens. But you want to keep in mind that not everything you see will be passed on to the progeny in the successive generations. So if you see something spectacular and you save seed from it and you don't see it the next generation, that is probably because it had more of an environmental influence on whatever it was that you saw rather than a genetic influence. And I didn't have a good example of tomato in that, but I do a lot of brassica work in my field. So this is actually a cabbage example, this photo here, and this is a Salix seed pods on a green cabbage, and I had never seen that before. I'd never seen 3 pods. Normally, what you normally see is this, just a single pod. And so this is a great example. I am quite sure that this was an environmental effect more than a genetic effect. So I wasn't anticipating getting 3 times the seed yield if I saved all my seed from that plant. So good to temper your excitement a little bit, but also good to keep your eyes open for things that are new and interesting. So now I'm going to take you a level into the genetics. So keeping in mind that these are examples that are very much simplified to hopefully make this easily understandable and to get the concepts across to you. But in reality, this is unusual, if not totally impossible, but you're not generally going to see genetics that are quite this uniform. But this is the idea. So each of these, the big A, usually in genetics, big A stands for a dominant trait and little a stands for a recessive trait. So we're looking at 2 genes here, the A gene and the B gene. These can represent anything. We're just using them as an example. So when you have a plant that's got all little a's and all little b's, all you're going to get for your gametes are a's, little a's and little b's. Same thing with when you have all big A's and big B's, the only gametes you can make are big A's and big B's. So all of your offspring are going to look exactly like your parents, and they're going to have the exact same genetic makeup. So if we were going to make a cross or hybrid, and this is how you would start a breeding project. If you had 2 tomatoes that you were interested in crossing, you would cross the 2 of them. And then you would get your F1 generation. And now we've got a big A and a big B and a little a and a little b. And this is important in how it plays out in the next couple generations. Because now, instead of just having the ability to have a big A, big B, and a, little b gamete, we now have 2 new gametes that we can form. Big A, little b, and little a, big B. So this is our F1 plant. Here it is over here. This is what it looks like. It's going to be somewhere in between our parents. And then if we took this plant and we self-pollinated it, we come down to all these new different kinds of genotypes that we can see in the F2 generation. And this is the Punnett square here, and the ratio here on the bottom are showing you the same things. These are the genotypes that this plant is capable of making. And this is why in that F2 generation, you're going to start seeing a fair bit of segregation, and you're going to start seeing new phenotypes popping up because you have all these new options of genetic combinations. So this is kind of a lot of information. If this is not something you've seen before, I don't want you to worry too much about understanding all of it. But the idea that I'm trying to get across to you on this slide is there's lots of new possibilities. And keep in mind, this is only 2 genes we're talking about, and there are tens of thousands of genes in a plant. So you can imagine that with all that opportunity for new combinations, you're going to get a lot of new and different phenotypes when you start growing out and saving seed from that. So that's why when you save seed from a hybrid, it doesn't grow true to type. Now, there can be a little bit of an exception here. It depends on how different these parents were. So if these parents, parent 1 and parent 2, were very phenotypically similar, but with different genotypes, let's say for disease resistance or salt tolerance or things, when you start growing that out in the F2 and F3 generations, it will probably still, if both parents were red slicers, you're still going to see red slicers. You're not going to see a lot of visual diversity there. So this is another representation of that same concept. And this is really neat. Again, a lot of information, so feel free to come back to this. In the F1 generation, if we had our parents who were big A, and little a, sorry, big A, big B, and little a, little b, we're going to take that down a notch and actually just look at A because it's easier to do than looking at two genes. And so when we make that first cross, all of our genes are now big A, little a. Again, we are dealing with a natural dynamic system, so this is nothing is 100% in nature, so just bear that in mind, but just to get the concept across. So if we took that plant and we selfed it, so we took that big A, little a, and we selfed that plant, now we have the ability to make big A plants, big A, plants, and little a plants. And this is the ratio that we're going to see those plants grow true in that generation. So 50% of those plants are either going to be, homozygous now, which is big A, or little a. And now the only gametes that those can produce are big A's or little a's. So all of the plants, if we self that again, all of the plants that have big A, or little a will produce big A, and little a plants. So in every generation of selfing, you get 50% increase in homozygosity. So if we take that F2 plant and we self that again, now in our third generation, we've got 75% homozygosity. So that means, again, we've increased our proportion of big A, or little a plants. And then we do that again, we self those plants, and now we've got 87.5% of the plants in our population are either big A, or little a. So this is great for breeding strategy, and as you're locking in those genetics, as you're creating more and more homozygosity, you're creating more and more stability in your genetics. So by about the fourth and fifth generations, you've got a very stable genetic combination. So with that, I am going to hand it over to Julie and let her talk about some variety maintenance and growing. Feel free to come back with your questions about genetics at the end.
Great, thanks. Whoops, I seem to have skipped. So I'm going to talk a bit about how you might go about choosing the varieties you're going to select or save seed off of, some of the field management and considerations there, and seed harvest. When you go to choose varieties that you want to save seed on, if you're like us, you probably have more than a reasonable number of varieties growing in your backyard or farm. So you want to kind of be selective about which ones you would like to focus on for seed saving. And those might be ones that you want to turn into a breeding project, or they might be varieties that you really want to maintain as is, and save seed to make sure that that variety stays available. So first you want to look at adaptation. A lot of these things are the same characteristics that you would look at in deciding whether to grow a variety, either commercially or for your own consumption. Adaptation to your climate. If your climate is extreme, here in Wisconsin, we get pretty far north. And so you might want to save seed because you can't find very many varieties that are adapted to a northern climate with a very short growing season. Make sure that when you're deciding about varieties that you consider how feasible the logistics are. So don't start with many varieties that you want to save seed on. Start with one or two and then work up as you get used to some of the logistics of growing seed rather than growing tomatoes. You want to make sure that the varieties that you are saving seed have good disease resistance to the main diseases in your area. And Dan will talk later about seed-borne diseases, which you have to be particularly careful of. But in general, you want to try to identify varieties that have good disease resistance. Sometimes that doesn't exist, so then you might need to modify your management to try to reduce disease in the field. You might decide to save seed on a variety because you can't find it somewhere, or you can't find enough of it for your needs. If you have a variety that you like but is available in a lot of different places at a reasonably low cost, then you might want to buy that variety and save seed of one that's harder to find. And when you're deciding to save seed, you want to keep in mind all of the different traits that you're looking for in a variety and see what already exists out there and what you might want to either have your own variety for, or you have a particular variety that either meets your market needs, maybe you have a very unique market. So kind of focus on varieties that are harder to get elsewhere, or that you particularly like when you're saving seed, because it is work, but it's very rewarding when you have your own variety and you know that you'll have seed of that. So then going to where you start, if you want to select varieties or source seed for then starting a seed saving project, variety trials that you've done on your own farm are often the most useful source of information. So you can identify varieties that do well in your environment. Often the seed catalogs, they do a very good job of trialing, but it's not on your farm. So you want to make sure that you trial things first. You might have heirlooms in your family or your friends that are unique. There are also a few interesting communities that if you want to get more involved in seed saving, you could kind of get involved with some of these groups that are very avid seed savers and amateur breeders, professional breeders. The Open Source Seed Initiative has a lot of professional breeders who are releasing varieties to encourage people to continue breeding and saving seed. The Dwarf Tomato Project is a group of amateur breeders who have done some really interesting work with small tomatoes for small growing spaces. Seed Savers Exchange obviously has a lot of heirlooms that might be interesting, and many smaller organic seed companies or mid-sized organic seed companies sell varieties where seed saving is perfectly legal and encouraged. So you probably want to check some of those out if you're trying to find varieties where you want to save seed. Sorry, it's not advancing.
Oh, you okay? Oh, there you go.
Oh, there we go. Sorry. So field production is similar to your tomato production. There are a few things to keep in mind when you're producing seed that is different than when you're just producing tomatoes to eat. Seed production always, you need to be more careful about diseases that might get transmitted from one generation to the next. So that means sourcing your seed from a reliable source so that you know that you're not starting with seed that has pathogens on it. Make sure that the transplants you move from your greenhouse to the field are clean, and then really make sure that you keep up with in-field sanitation, cleaning off dead leaves, and roguing of any plants that are showing signs of disease early on. For good quality seed, you want to pay good attention to the crop. Takes more eyes in the field to make sure that the seed crop is healthy. You probably will do a similar spacing for tomatoes, although if you typically get some foliar diseases that are exacerbated by high humidity, you might want to space it out a little bit more. And trying to trellis and prune leaves and branches or suckers as they start filling in can help reduce the potential for disease transmission. Some foliar diseases are not transmissible by seed, but they will reduce the quality of the fruit and potentially reduce the, you've put a lot of effort into making a seed crop, so you don't want to end up with very few fruit because you have a lot of foliar disease. So a few other considerations when you're growing a seed crop. There are a couple things to keep in mind when you want to maintain a variety as is, or maybe improve its adaptation a bit to your area, but otherwise keep it the same. First, you need to make sure that your population size stays big enough to maintain the intrinsic variation of any variety that you start with. So if you're starting with a modern variety, that may not be as much of an issue, a modern self-pollinated variety, but historic or heirloom self-pollinated varieties often are what we call population varieties, where individuals are genetically different. Even though the variety as a whole looks similar, there's still genetic variation there. And if you reduce it to too few individuals, you can start to lose genetic variation just by chance because you don't have enough individuals to maintain that in the population. You do, however, need fewer plants in general than a cross-pollinated crop like maize, for example. You also want to try to identify any off types, things that might have resulted from a cross. You know, if a bee pollinated your tomato plant with your neighbors, the next generation might look different. And so you can check that things are true to type. And having enough plants helps you see what the true type is and identify any off types. So for heirlooms or other more diverse varieties, probably you want to shoot for 20 to 50 plants. You don't need this to prevent inbreeding depression, but you need this to maintain the diversity in that variety. For newer varieties, you probably need a lot fewer plants because they're starting more as a pure line. Like Lori said, the individuals will reproduce true to type. So if they're all identical to begin with in the population, you don't need as many to maintain diversity. Sorry, it's slow advancing. So to prevent cross-pollination with another variety, which is one thing that can cause your variety to have off types in it or drift away from what it was originally, you want to make sure that even though tomato is a selfing species, you avoid chance cross-pollinations because it can cross-pollinate with other varieties. So the general recommendation is to have 75 feet between varieties for more modern varieties and 150 feet between two different varieties for older varieties. This is because of the flower architecture, which I'll touch on in a minute and Lori mentioned earlier. You can, if you want to, it's more work, but if you would like to save seed from more varieties and you don't have enough space for those isolation distances, you can protect the flowers from cross-pollination using bags, and then the varieties could be right next to each other. This takes a little practice and some skill in observing when the flowers should be protected, and there are resources to help you learn that if that's something you would like to do. You probably want to start saving seed in a simpler way with one or two varieties, and then if you really catch the bug, you can start saving seed of a lot of varieties using some hand pollination or bagging of the flowers. The other thing that can affect isolation distances besides the floral architecture is the production system. If you have more pollinators around, you're more likely to get cross-pollination. So organic production systems may have more pollinators just within the production system, which is a good thing. It also may increase the risk of outcrossing. High tunnels often have fewer pollinators than the field, although we plant flowers in our high tunnel to encourage beneficial insects, so we have almost as many pollinators inside as outside. So your production system may influence the actual isolation distance that you need. Obviously, the recommendation is a general recommendation that doesn't hold true in all situations. The floral structure Lori mentioned earlier. Heirlooms are more likely to have an exerted stigma, so they're more likely to have cross-pollination. Modern varieties are more likely to have the anther cone completely wrapped around the stigma, so they are more likely to self-pollinate first. So you need to be aware of that, which type of variety you're trying to save seed on and what its flowers look like. Potato leaf varieties are slightly more likely to have the exerted stigma than regular light leaf type. The easiest way to tell is to look at the flowers when you're growing the variety. So for seed harvest, the main thing to remember is that the seed is maturing inside the fruit, and when the fruit is totally ripe, then the seed is fully mature. So it needs to be really dead ripe, more than what you would want for eating. The seed maturity influences the quality, the germination, so you want to give it the best chance it has to fully mature. You also don't want to harvest fruit that might have some rot in the field or let it rot after you pick it. So there's kind of a narrow window of where to get optimal fruit quality for seed production. Many people, if they're doing this on a small scale, may want to use the fruit as well as save the seed. So you kind of can compromise a little and not pick it overripe, but you want to get it really ripe. And if you're going to use the fruit for something, tomato sauce or salsa, as well as saving the seed, then you have to be very careful about food safety precautions as well. So when you harvest the seed, there's a few different ways of doing that depending on the scale. On a very small scale, you can cut it open, scoop out the pulp with the seeds, put it in a little container. On a larger scale, you may want to push the fruit through a mesh to try to get rid of some of the pulp before it goes into the fermentation process. You can also put it all in a bucket and squash it to the point where it can start fermenting. So there are different ways of getting the seed extraction process done. You just don't want to wait too long before you do it so that the fermentation process can start before you have a lot of rot in the fruit. And the different scales will depend on how much you're producing per batch, really. If you have a lot of small batches, you're probably still going to be using a smaller scale method than if you have one big batch, even if you're producing the same total quantity of seed. So with that, I will turn it over to Lori again to talk about fermentation.
Great, thanks, Julie. So fermentation is a really neat and fun part of tomato seed production. You don't have to do it, but I have found it's definitely the easiest and most effective and surest way to break down the placental tissue around each seed, and that's sort of that gel tissue that you find around all the seeds. It's delicious, but it also has germination inhibiting compounds in it, so you very much need to get it off the seed so that you'll have good germination and quality seed. And you can do that using fermentation. You can also do that during using rubbing or spraying. I've found you have to do some sort of fairly aggressive abrasion to get that gel off, and it's quite a lot of work, and that generally you're not going to be as effective at getting it off as fermentation is. So I was very intimidated the first time I did tomato seed fermentation, but have found it's actually very easy and fun to do. And as well as getting rid of that germination inhibiting gel, it loosens up any pulp that's clinging to the seeds and makes it easier to remove. And it can destroy some diseases. Dan will touch on that in just a moment. So Julie said the easiest way to do it is to remove the inner pulp from each tomato if you can. If you're doing a small enough scale, then you just have less pulp to deal with later. If you're doing a larger scale, it's going to be easier for you to mash whole fruits. There is some disagreement about whether to add water or not when you're fermenting. I do not add water. I only add it if I'm doing a tiny bit of seed and I just don't have enough gel and enough liquid to really get the fermentation going. And one thing you want to think about is what Julie was mentioning about wanting to use fruit that's really dead ripe. Another reason to do that is because you have a lot of sugar in those nice, super, super ripe fruits, and that sugar aids the fermentation. So if you're using underripe fruit, you not only risk having not fully developed mature seed, but you also risk not having good fermentation because you just don't have enough sugar to get it going. So this photo you see here, this is some breeding population fermentation that I'm doing. So I wanted to show you can do fermentation on a very small scale. These are quart mason jars. I've got some pint mason jars in the back. So you don't have to have a ton of fruit or a ton of seed to do this. So I have done my fermentation in the greenhouse. You can also do your fermentation in your kitchen or anywhere that's warm. It wants to be warm for all of that sugar to get going and break down, preferably somewhere around 75 to 85 degrees. If it's above 90 degrees, you risk it getting too hot. So you do want to monitor that. And you can see here, I put shade cloth up on my greenhouse. This is pretty much in the height of summer, and I don't want it to be 110 degrees in here while I'm doing this. I allow fermentation to go for about 3 to 5 days, And determining when fermentation is done is a very individual thing. Some people disagree or have different ideas about how they decide it's done. The general best way I found to really get a feel for it is take some seed out and feel it. Rinse it off and feel it between your fingers. And if you can still feel that slippery gel, it's not done yet. You do want to make sure you're not leaving it in too long, because if that gel breaks down and the seeds get nice and soaked and wet, they can sprout. So you want to keep an eye on them and make sure you're not seeing any sprouting in your seeds. If you are, get them out right away and don't let them sit in that fermentation juice any longer. So I generally stir once a day, maybe twice a day as I'm walking through my greenhouse. That aids the fermentation process. You can just stir all that pulp and any mold that starts to grow on the top, you can just stir that back in. Some people consider the fermentation done when they stop seeing the bubbling on the top. So you can see a little bit of it here. You start to see this separation, and maybe you'll see some white mold forming on the top. You'll notice active fermentation is very bubbly, and then it will kind of calm down. Others will stop when there's a pretty thick layer of mold covering the top. So you'll just have to get a feel for it if you haven't done it. Again, just take some seeds out and feel them, and you'll figure out with your situation and containers and how warm it is, you'll figure out when it is done. So when it is complete, you're going to decant and rinse and dry your seeds. So a couple more pictures. This is Andrew Still from Adaptive Seeds, and he's doing the same thing that I did. This is actually the picture that inspired me to ferment in little mason jars. And here's what it looks like if you're doing a larger scale. You can use 5-gallon buckets, you can use trash cans. It all depends on what your volume is. And this is also a photo of decanting. So when your fermentation is complete, you're going to fill up whatever your vessel is with water and stir it around, wait for a minute. The fermented seeds will actually settle in the bottom, and you'll have all this nice pulp that you can just pour right off the top. And that first pour can be pretty intimidating because you have a lot of pulp coming off, and especially if you're using an opaque container like this, it's hard to tell when to stop. So you just pour until you're to your comfort level, fill it back up with water again, and keep pouring it off, and eventually that water will just run clear, and you'll end up with beautiful clean seed on the bottom. And then once you do, you want to dry it out. Again, this is our little greenhouse, and because I was doing very small scale, lots of populations, but small amounts of seed, I use window screen. This is a great way to maintain aeration. I've got a box fan going on here to make sure I'm keeping my temperature down and I'm keeping my airflow up. Each of these pieces of blue tape, that's how I've got. So that's the cross, and those are my labels. And I have not ever had, I keep my fan on fairly low to medium, so I'm not blowing seed around, so I'm not getting seed from this pile blowing into this pile. If I do happen to end up with any seed that's in the middle, I just toss it, or I keep it for my own personal garden. Anything I'm not 100% confident is the cross that I want. I have also found that if I go in and I break up these clumps when the seed is about 70% dry, it breaks up easily. If I wait until they're dry, they all just stick together, and it can be really difficult to break them up into individual seeds. So I just go in with my hands and I'll kind of lightly push them against the screen, or just pick up a little bit and crumble them in my hands, and I found that they detach from each other quite nicely. This is where I learned to ferment tomato seeds on window screens. This is another farmer, Beth Braskerchek from Utah, and she stacks her screens. And again, because you've got these frames, they're not sitting right on top of each other. And if you're limited on space, that's another way to do it. And then once you have dried your seeds, you want some storage considerations to take into account. Seed is alive, so even though we dry it down and it's essentially dormant, it is still breathing and respiring. So conditions that are cool and dry and dark will help keep that seed breeding to a minimum. You want to have an area to store in where you've got minimum fluctuation of both temperature and humidity. And then you want to think about whether, if you're confident that your seed is nice and dry, and you want to dry it down to about 5% moisture, then you can put them in airtight containers. If you're worried that your seed is not quite dry enough, you can put desiccants in those containers with them. Or if you've got a nice stable storage environment, you can store them in cotton muslin bags or paper coin envelopes, or something that's a little more breathable and responsive to the environment. But you want to make sure you're protecting them from pests as well. Underground storage is pretty ideal because it's a nice way to maintain pretty constant temperature. So this is an example of some friends of ours from Sierra Seed Cooperative. And with that, I will turn it over to Dan to talk a little bit about seed disease.
Okay, thank you, Lori. This has been a really nice infomercial about saving seeds. Julie and Lori have presented a lot of good information, and I hope you're enthusiastic about saving seeds. But I'm kind of give a little disclaimer here. I'm kind of like the guy at the end of the commercial who talks real fast about side effects. So when you're saving seeds, you do need to be careful, and Julie touched on this, that some diseases can be transmitted through seeds, not all of them. So what I recommend then is to know which diseases in your area may be seed transmitted. So for example, if you're in the South, you're probably more likely to have insect transmitted viral diseases. If you're in a arid area, you're less likely to have a lot of foliar diseases. So you need to know what diseases you have. And I recommend to as you're starting out and you see diseases and learn to recognize and problems, I should say, get an official diagnosis of what that problem is. And it always sort of amazes me that when I'm talking to people about diseases on plants, and they will go to any amount of trouble, expense to solve that. And then when I suggest over the phone, for example, to get an official diagnosis, they say, no, I don't need to do that. So, but I'm here to encourage you to get an official diagnosis, and never save seed from a diseased plant or a plant that you might suspect of being diseased. And certainly it's possible, if at all possible, you don't want to save any seed from a plot or an area that has any disease, but certainly you would want to save from an individual plant that had been diseased. Okay, so what I've done here is to have these 3 photos of tomato problems, and ask whether you think that any of these individual problems can be transmitted through seeds. So the first thing would be to identify those. And again, I encourage you to get those identified if you don't know. These are late blight, blossom end rot, and anthracnose. So, but that in itself doesn't tell you whether there is seed transmitted. Certainly you wouldn't want to save seed from any of these tomatoes, but if you had any of these diseases or any of these problems in your plot, how important would it be to be transmitted? So the one on your left then is blossom end rot. This is a calcium deficiency. It's not seed-borne. Certainly I wouldn't save seed from that, but it's not going to be transmitted through the seed if you had a few of those tomatoes in your plot. The middle one then is late blight. This can be seed-borne. It's not real common, but certainly you wouldn't want to save that seed. But the one on the right then is anthracnose. This is commonly seed-borne, and certainly if you had this in your plots, in your garden, you would try to avoid saving seed from that at all, because this can definitely be seed-borne. So if you are unsure, you don't have to send every lesion, every fruit off to a diagnostic lab, but send representative ones as you have questions to an official diagnostic lab. And what I have photos of here is on the left, bacterial spot, which is very easily seed transmitted. In the middle, this is edema, a physiological problem related to water. It's not infectious, and it certainly will not go to seeds. And then finally, on the right is powdery mildew, which is a problem on tomatoes, but is not seed transmitted. So that's just my plug for getting things diagnosed if you're unsure. In general, then, there's two ways that disease pathogens or microorganisms that can cause disease, there's two ways that disease pathogens can be transmitted through seed. And the first way is there may be disease lesions on the surface of the fruit. And down below, you see a photograph, an ugly photograph of bacterial spot on tomatoes. And certainly, again, you would not save this, but if there was some of those in the plot, it'd be easy to miss some very small lesions, and you could pass it on that way. So you can imagine taking those bacterial spot tomatoes and slicing them open, and it'd be very difficult to avoid transferring the disease from those lesions, the pathogens from those lesions onto the seeds. So you would want to avoid that. The second way that pathogens are transmitted into the seed then is a systemic infection. And the photograph on the right then is Fusarium wilt, and you can you can tell that because it's got kind of a one-sided wilt. It's wilting one side of the leaf more than the other, and that's because the vascular system, the system that carries water through the plant, is plugged partially. So although fruit on that plant would not show the disease, it's possible that the fungal spores would go right into those seeds. So the two ways, lesions on the surface of the fruit and then systemic infections could cause a seed to have pathogens or be disease transmitted. So here's bacterial spot, external contamination, and again, you'd want to avoid saving those seeds. Just some examples here. This is bacterial canker, which also has lesions on the surface of the seed. You can see here, or on the fruit, and certainly if you slice that open, you can see the seeds here. If you slice that open, it'd be very possible for the bacteria to go right into the seeds. This is bacterial canker of tomato, very serious disease. But bacterial canker, as well as causing lesions on the surface of the fruit, also has a wilt phase, as shown here. So this is a wilt, and it's wilting because the vascular tissue, the plumbing there, if you want to speak about it that way, the plumbing of the plant has a disease, and because the bacteria is in the plumbing, then that means it can get inside the seed. So bacterial canker is one disease where you can have contamination inside and outside of the seed. All right, and then here's just a close-up of Fusarium wilt. You see that one-sided wilt it's one side of the leaves. This is typical, is affected, or one side of the plant is affected more than the other, and that's because, as a professor of mine used to say, the plumbing in part of the stem is blocked off. All right, so how to save how to save healthy, how to do the kind of stuff that Julie and Lori were talking about and do it healthy. So certainly you'd want to avoid saving seed from a field with the disease plants. And certainly you certainly want to choose tomatoes. And I think if you're doing a small scale, you could choose tomatoes that are individually healthy. If you're a large scale operation, it's easy to see how you might get a disease when thrown in there. But on a small scale, it's easier to sort those out. If you have a small enough operation, you might want to actually surface disinfect the fruit with a little bit of bleach or ethanol or whatever's allowed in your operation. And then Laurie was talking about fermenting, and so there's various ways, as Laurie spoke about, to ferment seeds, but and it's certainly fermenting it won't solve all your problems, but there is some good evidence that seed that's fermented will help to lessen the disease problems. If you think about it, how that may be true, because in general, if tomato falls on the ground, and naturally it probably ferments in the fruit. So fermentation is okay. If you don't want to do that, that's all right. But I certainly have no problem with using seed fermentation. I'll have a little bit more information on the next slide, but it's also possible that if you're worried about disease, to hot water treat or to bleach your seed. And of course, you want to make sure what is allowed by your certifier. But after you've harvested the seed, then to go in with a hot water treatment or a bleach. The hot water treatment then would be for pathogens inside or outside of the seed, and a bleach treatment would be just sanitizing the outside of the seed. And again, you'd want to check with your certifier. And so for more information about treating seeds, and I would encourage you, don't go out and heat treat your seed, for example, without trying a small amount of it first, because it certainly will be possible either with a heat treatment or with a bleach treatment to ruin or very much lower the germination of your seeds. You'd want to try a small portion of it first. But if you want more information, here's an article, and Alice has given a good link to that article there, and she talks about it, and it talks about it from an organic standpoint as well. So that's all I had, and I'll hand it. Well, I guess I had one more slide.
Okay, thanks a lot, Dan, and everyone. We're going to begin the question and answer period, and we've been getting a number of question and comments throughout the webinar. We have a couple tips and comments as well from our audience. But first off, if you are making a hybrid tomato, what traits of the plant are carried through to the next generation by the male versus the female plant? So in other words, if one variety is playing the role of female, it will have the same shaped fruit of that female variety. Can anyone talk about how you might know that? Everyone has to unmute themselves if you're not.
Yes, yeah. I don't know that there are systematic differences between using a variety as a male versus a female. The one thing for sure is that the cellular organelles, the chloroplasts and the mitochondria, come from the female line, but that doesn't necessarily affect what happens to the fruit shape. Typically, a lot of characteristics that are quantitatively controlled genetically, so many genes and the environment influencing the phenotype. You see something that's kind of midway between the parents. For hybrid tomatoes, often the combination can be used to maybe combine disease resistance and flavor, because you'll get the disease resistance from one parent and the flavor from another faster than an OP, although you'll still have something that's kind of in the middle. You won't necessarily get the best from both parents, but it's not always predictable. And I don't know that there are characteristics that are better as the male or the female for the plants. There are males and females, or there are varieties that work better as a pollen parent. They produce more pollen, or work better as a female parent because they set better fruit after cross-pollination, but I don't know that that affects the progeny. And Lori, you're welcome to add to that.
Yeah, the only thing I have to add to that is I generally will make, if I'm making a cross, I'll make it both ways. And because sometimes, as Julie said, there's a lot of unpredictability in how all those traits play out and recombine. So I'll do both, because sometimes one does sort of magically emerge as a better mother or father.
Okay, thank you. Here's a comment. Someone recommends considering the plant yield and seed yield of a variety when you're determining the population size. So for example, paste or meaty beefsteak tomatoes might have a lower seed yield per fruit and require more plants if you want to produce a lot of seed. And yeah, so varieties with lower yields may need more plants. So that was a good point. Thanks for that.
I think that's certainly true for when you're looking at how much seed you want to produce. It's the number of individual plants that matter for the genetic maintenance. But for seed yields, it is the yield of the plant as well.
Okay, yeah. So we have a couple of people asking about, why is it so important to prevent diseases while the plants are growing? And why is it so important not to save seeds from diseased plants? Because you're going to ferment them anyway, for example. Can you talk more about that?
Okay, so why prevent disease while they're growing? And one of the things that Julie mentioned is just from the standpoint of production, if you want to produce, you're producing seed, you want to produce as much seed as possible. And certainly if you have foliar diseases, even if they're not the kind of diseases which are transmitted through seed, you're probably going to lower your yield. And you may lower, if you have diseases, you may end up that you've lowered the germination of the seed. But so, for example, a disease like early blight, I don't know that to be a problem with seed transmission, but it would lower the yields and certainly make smaller fruit and whatnot. But bacterial spot is very easily, you'd want to reduce, if not eliminate, the possibility of bacterial spot, because once that's in there and you have it in your seeds, then you've passed it on to all the progeny. So that's why I would. I just see so many cases where it looks like somebody has given themselves problems by the seed. Let me further say that a lot of times when you produce transplants from seed, you do so in a greenhouse and water them overhead. And if you think about those transplants in a transplant tray, they're right next to each other. They're watered overhead every day, maybe multiple times a day. So even if you have just a few transplants with disease on them, it can rapidly spread throughout those transplants and maybe even into a different variety.
Okay, thank you.
And I would just add, I want to add to that fermentation is definitely not a catch-all for all diseases. And so the best way to prevent any disease transmission is to save seed from healthy plants that don't have disease. So yes, you are fermenting. Yes, that can kill some disease, but it's definitely not going to kill all disease. And even on a disease that will die with fermentation, if you're saving from a heavily diseased plant, I wouldn't count on fermentation to get rid of all the disease.
Okay. Do you know where someone can find tips for hand bagging flowers or resources to learn how to do that?
I think there are a couple webinars on tomato breeding online. There's one from Ohio State, I know. And then I don't know, Lori, if OSA's guide includes that. I think it does.
Honestly, I can't remember. I think it touches on it, but I wouldn't say it's a
right. It's not step by step.
I know you can do like a YouTube search, and there's a number of videos, pollination videos on.
Okay, I'll look after the webinar too and
strategy.
see if I can find anything to send to that person if they're interested. I
think the tomato breeding one we did last year also has a little bit on that Jim Myers and Kara Young did.
Yeah, so if you search the eOrganic YouTube channel and you look under tomato seed, you can probably find what Julie's talking about. And I'll also try to find something for that person too. Okay, here's an interesting question. What about varieties that seem to consistently sprout even at day one already? We work on a small scale using the mason jar method, and we had sprouting as a major problem last fall with one variety, which we had a customer who wanted a huge amount of it and isn't readily found elsewhere. So they had a problem with sprouting after 3 days, so they tried 2 days, and then they even had a problem after 1 day. It was very frustrating. So do you have any thoughts or advice on this? They found that out of 30 or so varieties and many batches of each, this one variety was the only consistently problematic one with sprouting seeds.
I have noticed that too with just one or two, their breeding lines, but I don't know that it's something that you can manage with changing the fermentation. I have not figured it out yet, but I have noticed the same thing with just a couple lines.
Should one, using the fermentation process keep the lid open of the jar, or would it hinder it to put a loose lid on top of the jar?
I don't think it would hinder it. I keep mine open just because I'm doing them in a greenhouse, and it doesn't matter. I forgot to mention you will attract fruit flies. So if you're doing it in a space where that's an issue, yeah, I would put a screen over it, or you could rubber band a piece of cloth over it. Just, it should still breathe a little bit, but that should be fine.
Yeah, we just put a little plastic lid over ours that goes with the small plastic cups that we use because we're doing it often in a closed environment, a lab. It will blow the lid off if you close it tightly, so don't do that.
Okay, good tip here. Here's another tip. Someone has a comment about how to defuzz the tomato seed, which makes them easier to handle when they're sowing.
Yes.
So he recommends placing dry seed in a double layer of nylon hose and tying off the end, and then putting this in a tumble dryer with no heat and a couple of tennis balls, and check after 10 to 20 minutes until the desired effect is achieved. So that has worked for him. I don't know if you have any other tips about that.
Very cool. I would love to try that.
Okay.
I've also heard people, we do small batches, so we don't have as much issue, but people putting them in a fabric bag, or the pantyhose would probably work too, and put a small piece of chain in there. So just a little metal chain. And then if you rub it, it'll take off some of the fuzz.
Interesting hacks here. Let's see. Here's someone who's based in the UK, and he's heard of people treating seed with vinegar to prevent viruses. Is this anything that you have experience with?
No, I've never heard of that. And just my kind of gut check is I don't think that would be very effective. But I'd have to do some research on my own to know that. I don't know if Julie and Lori have ever heard of that, or if they know what vinegar would do the germination.
I don't have any experience with it either.
I don't think it would be effective with viruses. Sometimes people use hydrochloric acid, which is also an acid, but that is not allowed in organic. So I don't know that it would affect the germination because hydrochloric acid is also acidic. But just from what I know, I don't think it would work against viruses.
I would think viruses would be internal in the tissue.
Yeah.
Okay, let's see. Can you speak maybe, Dan, about how somebody would get an official diagnosis of plant problems?
So I'm in Indiana. I'm not Indiana now, but at Purdue we have a diagnostic lab. We on the campus, West Lafayette campus, the Plant Pest and Diagnostic Lab. I think most states that have a land grant college. Lori, that'd be you in Madison, I would guess, right? But most states have a land grant college. And that's where I would first look to. If you didn't know where that was, I would contact somebody like me and ask help. And people do contact me about that, and or go on the internet and look for the internet, the web. I don't know, Alice, is that something that maybe we could publish a list
of land grant universities? There must be one with the plant diagnostic labs. In fact, we may even have something like that in the Organic Seed Resource Guide online. I'd have to double check, but that's something that you can easily find online by just simply typing that into a search engine to find the land grant university and then type in your state. I know here we've got one here in Corvallis, Oregon, so OSU. So.
We have the Midwest Vegetable Production Guide. I know we have a full page or pages of diagnostic labs. Go ahead.
Yeah.
I was going to say it's also a good idea if it's not something you're real familiar with, you can always call and ask, and they always have resources on how to take samples and what you need to send in, and sometimes you can even talk to someone and explain what it is that you're experiencing, and they can kind of help guide you as to whether that's something you would want to take a sample and get a diagnostic.
That's a good point. And they might say, gee, if you're sending in that, for example, if you're sending in for some fusarium wilt, they probably don't want you to send in the leaf if that's what you suspect. They probably want a portion of the stem or maybe even the roots. So, good point.
Okay. Here's a comment saying, thanks for saying check with your certifier regarding rinsing seed in bleach. It really is important if you're going to use any kind of substance that you have any questions about to check with your certifier. So somebody's reinforcing that, and I think that's important.
Yeah, the concentrations as well can be different for sanitation versus something you're using. So definitely check, even if you've used bleach in your greenhouse to sanitize, check that it's okay on seed.
Okay, yeah, there's quite a few people asking about where they can find their local diagnostic lab, and I'll see if there is a list online. Maybe we can post that along with the webinar recording. But definitely a search engine, I think, would be a good start.
Yeah, and usually if you call somewhere, they can refer you to other places, because some seed pathology is somewhat of a specialty, and so not all diagnostic labs will do seed pathology if you want to get seeds tested, and sometimes you'll have to send those farther away. So sometimes you do have to do a bit of sleuthing and get recommendations and kind of follow a somewhat circuitous route depending on where you are.
Find the right person, yeah. Okay, here's a question about how hot water treatment affects the germination rate or longevity of the germination rate, recognizing that tomato seeds can be viable for 20 to 30 years.
I don't. I have not done that kind of research. What you posted, Alice, was Emily Gatch of I guess WSU. She may have done that. Yeah, right there. And then I know Sally Miller at Ohio State has done that work. So I'm kind of following in their footsteps. And if done in that way, we have done this in our lab, and if you follow that very closely, I don't think you'll hurt the germination at all. That's been my experience. But I can't tell you how far outside of that will start to ruin the germ. I haven't done that research, so that's why I just kind of follow what others have done and tread lightly.
Yeah, if you get it too hot or for too long, it will affect the germ and the longevity, because it kind of starts the seed germinating. But I think the best thing to do is to find a guide and to follow it carefully.
And my limited experience and gut reaction is hot treat before you're going to plant, but don't hot treat and then store.
Uh-huh.
Yeah. Yeah.
Okay, great. Here we have a group watching here, and they have lots of questions. Here's one of them. We see a disease that maybe resembles your pictures of bacterial spot, but the spots are the size of a pinhead, dark black and very superficial. Might this be the same thing, or perhaps a different disease?
So just a guess here, but I would guess that's bacterial speck. So it's related in the sense that it can also be seed-borne. Certainly, I've been able to find that on transplants before. I can go to some of the big box stores where they have transplants out and find that, and bacterial spot. But without going to a diagnostic lab or doing some lab tests, I wouldn't know for sure. But my guess is that would be bacterial speck. And I apologize that us pathologists are so lame with our disease names.
All right. And another question from that group. Why avoid, let's see, where did that question go here? Okay. Why avoid fruits that are so ripe that they verge on spoiling? Isn't this just the beginning of the natural fermentation process?
It can be. It can also have other bacteria in there or fungi that you wouldn't necessarily want in your fermentation process. So there are good microorganisms and pathogens you want the pathogens you want to keep out. Most of the pathogens that would kind of start in rotting the fruit are probably not as much seed transmitted. They're probably saprophytic, or they eat dead tissue, but you still don't want a lot of additional potential pathogens in your fermentation process. The fermentation microorganisms, it's similar to any other kind of fermentation, whether you're making bread or beer, you have certain organisms that really drive the fermentation, and those are the ones you want in there.
Okay, we have another tip here. It says seeds are ready to be extracted at the breaker stage, so when there's a blush of color showing on the blossom end. If precocious germination occurs, harvest fruit at the breaker stage. So this sounds like someone who has tried this at home. So a tip, see if it works.
And I have actually done that as well. I took off a late fruit set and kept my seeds separate from my main seed harvest, and then checked the germination against each other, and they both were fine. So that can definitely work. I think you just have to watch your fermentation, because you may be low on your sugars. But that's a great recommendation to try for the variety that seemed to be very.
Okay, and we have one more recommendation from him, and he says, don't add extra water when fermenting. So I don't know, but sounds like he's had experience with this. So, okay, so here's a question about, okay, it says in the UK, it's very difficult to get seed to dry. When do you know that seed is dry enough to be put in sealed containers?
That is a great question, and one that can be kind of challenging. We have that problem. I'm out here in the Pacific Northwest in Washington State, where it's not often very dry. So you can do a couple of things. If you have a dry environment, you can bring them, especially if you have a wood stove, you can bring them in your house and get them somewhat artificially drier if you can't get them dry enough outside. And depending on how accurate you want to be, I often use a hygrometer, and you can buy them pretty cheaply online, and I'll put it in a sealed container with the seeds and see what it comes out at. That's not giving you a totally accurate read on the moisture percentage in the seed, but it gives you a fairly good idea. And I try to get mine down to around 30% relative humidity in that closed environment. That's what I was taught, and that seems to be holding true for the seed storage that I've done. You can also dry them down. Some people will use like a dehydrator, but you want to be very careful doing that, because if you get them too hot, you can kill them, or you can dry the outside too much, and then your seeds can have trouble imbibing water. Another option is to use a coin envelope. It's got not the peel and tape, but the sticky closure, and put that in a sealed container with them. Leave it for about 24 hours, and if you take it out and that sticky part is sticky, then your seeds are too wet, and if it's still dry, they're dry enough to store. You can also do it with if you have a crisp new dollar bill, and you put it in there for a day. If you take it out the next day and it's no longer nice and crisp and dry, that means your seed's too wet and there's too much moisture in there. So those are kind of the non-scientific ways to do it. I don't know if Julie or Dan, you have anything to add?
No, that's good. They also, you can, if you're having trouble drying it down, they do sell silica beads that you can use to absorb more moisture out of the seeds once they've been put in a container. And those can be, you can either get them to use once or the
Okay.
piece of rule of thumb would be that relative humidity plus temperature in Fahrenheit, and sorry that you're in the UK because I only know the Fahrenheit, relative humidity plus the temperature in Fahrenheit should be less than 100 for storage. So you want to dry your seeds dry enough and then make sure that you're keeping them dry.
Okay.
One that
yeah, and sometimes those desiccant beads actually change color, so they'll be like blue and red, and so you can, it's a visual indicator of they've absorbed as much moisture as they can, which can be kind of cool too.
Okay, here's a question about at what point will inbreeding degeneration be a problem from selfing generation to generation?
It won't be in tomatoes.
Right, inbreeding generally is not a problem in self-pollinated crops, but losing genetic diversity from having too few individuals in a population that started out as more diverse can still be a problem. So an individual plant, if you self it for many, many generations, you will probably be okay. Obviously, nothing is 100%, but in general, selfing crops have evolved to self, and so they've purged all the genetic baggage that can cause inbreeding depression in organisms that want to be outcrossed.
Okay, we have a couple people wondering about trisodium phosphate to remove the germination inhibitor, as is sometimes done with potatoes.
I don't think that is allowed in organic, but I can check.
Okay, yeah, I don't think it is either, but it's something that you definitely want to check before doing. Okay, let's see. We have time for maybe one more question here. Is there a difference in seed quality between the first trusses of fruits and the later ones?
I think the first ones may be more likely to outcross, but I don't know if the seed quality is different. Lori?
My gut reaction is not really. Depends a little bit on your environment. I will say if you are working with a tomato that has a parthenocarpic ability or tendency, meaning it will set seed without, it will set fruit without fertilization and without seed set, which is very helpful if you're in a cool climate and you want fruit production, but can be frustrating with seed production. And a lot of the tomato varieties that have come out of the Oregon State program do have parthenocarpic tendencies, and I have seen people be contracted to produce seed, not get any seed on those early flushes because it was cold, and be very frustrated. So that is something to keep in mind, and you'll get greater seed set as it warms up and you get more fertilization happening.
Okay, well, we're running out of time, but I'd like to thank everyone for submitting all these great questions. And thank you so much, Julie, Dan, and Lori, for coming today and telling us all about how to produce tomato seed. We hope very much that you can join us for the Organic Seed Growers Conference broadcast coming up in February. Thanks to everyone for joining us today.